All right, so we're going to talk about proteins in this presentation. So it is the most abundant organic molecule in living systems. Basically, your body is composed of mostly all proteins. They contain carbon, oxygen, hydrogen, nitrogen, and sometimes sulfur. There are some amino acids that have sulfur in them. They're very diverse, not only in their structure, but their shape and their function. Some are structural in your body. They are used for structural support. Some are for regulatory, contractile, protective, transport, storage. We've got some proteins are used for toxins or enzymes, or there are so many different types of proteins. And basically that structure of that protein is what gives it its function or contributes mostly to its function. Now, proteins are polymers. They are made up of amino acids, so their monomeric units are amino acids. And they do originally start as a linear structure, right? As we are, and we'll learn how we put that primary sequence together when we get into DNA, right? And how DNA works. So when we talk about some types and functions, we'll talk about enzymes. We're going to be doing some labs with enzymes. They are proteins that speed up the rate of chemical reactions. They do this by decreasing the amount of activation energy. So activation energy is kind of like that hill you need to get over to get things started. It's kind of like you in the morning, right? Once you can get out of bed, it's all good. You need that activation energy to get going. So what enzymes do is be like having a bed that just shoved you off, right? So it lowers the amount of energy that we needed to get started. Each enzyme is very specific to a substrate. What that means is it binds to a specific substance. That substance that it binds to is referred to as the substrate. An enzyme may assist in hydrolysis or in dehydration. So an enzyme could be catabolic or anabolic, right? And like I said, enzymes have up just a ton of different jobs that they do, but each enzyme is very specific to its job and specific to what it attaches to. So very, very specific. We also have some proteins that are hormones, and those are molecules that are important for chemical signaling between cells. We know hormones regulate specific physical processes like growing, development. We talk about going through puberty, or for us women going through menopause, those lovely things. And of course, reproduction. Proteins also provide structural support. Plants have several different structural proteins we have seen under the microscope, some cell walls. Those cell walls are made up mostly of cellulose, which is, we already talked about that with carbohydrates, but they do have proteins in them. We saw like the plasmodesmata, and there are other types of stuff in there. Actin and myosin are two major proteins that make up your muscle. And so they are important for contraction, muscle contraction. And then we got keratin, which is another one that's hair, nails. Those are all major components of your, basically your outside. Okay, so your skin is full of keratin. So lots of different jobs. Here's a table of just a few of those types of proteins and their functions. So we have digestive enzymes, amylase, lipase. Lipase breaks down. Well, let me start. Amylase breaks down starch. Lipase breaks down lipids. Pepsin and trypsin both break down proteins. We've got transport. We got hemoglobin. You know that transports oxygen around the body. Albumin is also found in the body. It helps to regulate pH. It's a buffer. It helps to maintain your blood composition. We have structural support. I already talked about actin. We have tubulin, which is a structural support part of the cytoskeleton inside our cells. And then we have keratin, which I talked about already. Hormones, insulin, thyroxine, which is important for your thyroid, insulin for regulating sugar levels, defense, all your immunoglobulins, your antigens, all those things, antibodies, those are all proteins. Contractile, we talked about actin and myosin there again. And storage, we could talk about lignin, which is storage proteins, and again, egg whites, which is an albumin. We actually saw that in our labs that we did when we were looking for proteins. So the lignin is a protein that helps with providing food for seedlings. Proteins have different shapes. We have this globular. I know globular is not a geometric shape. And it's like, how do you explain? It's a glob. It's all wadded up. It's like taking your fitted sheet and just putting it together, because how many of us can actually fold one of those fitted sheets? And so it ends up looking like a glob. All right. So glob, it kind of is a globe. Yeah, like your ball of fitted sheet. All right. So hemoglobin is an example of that. Hemoglobin, we'll talk a little bit more about that later. We have fibrous proteins that tend to be long and cylindrical, like collagen, helps hold your skin together. We have 20 different types of amino acids that are used to make all these proteins. And these different proteins have different types of arrangements that they're amino acids. Again, it makes them, each protein, because of the way those amino acids are put together, are very unique. So amino acid, when we look at the shape of our monomer, we have our amino group here. We have our central carbon, which is attached to our carboxyl group. So look at these two functional groups here. R is the side chain. Again, R means that it is what is different with each of the 20 amino acids. So we can have all kinds of different things attached there. That's like our, I wouldn't say it's our unknown, but it is our variable. It is what is different between the 20 amino acids. So like I said, our side groups on our amino acid, that's what we refer to as the R is your side chain, can made up of all kinds of different types of atoms. So, but we do have 20 different varieties. It's not like there's 100 different amino acids. There is only 20 different side chains, giving us 20 different amino acids. So here is a picture of those 20 different amino acids where the side chains are highlighted in that light blue. Those light blue regions are broken down into their different characteristics. We have nonpolar, and again, the amino acids themselves are polar in that they have those polar side functional groups, right? But when we refer to polar or nonpolar or negative or positive or neutral, we're referring to what is the characteristic of that side chain. So we have some nonpolar side chains. We have some polar uncharged. We have some nonpolar. We have some negatively, so they're polar as well, negatively charged, and some polar that would be positively charged. So they have different characteristics, and those different characteristics are going to give each of those proteins their different shapes. The chemical nature of the side chain determines the amino acid's nature, its structure, its function. Like I said, we've got the nonpolar or hydrophobic side chains. These are mostly hydrocarbons that have those nonpolar covalent bonds. I already talked about the polar. They are hydrophilic, water-loving. Our positive charge will also be polar, water-loving. Our positive will tend to have a basic pH. And then we have that sequence. The sequence is how we put that primary sequence is how do we put those amino acids together? I always think of it as like having a box with 20 different beads and you're going to make a necklace. And you have so many options of how to build that necklace with those 20 different beads you could use. And so we need a set of instructions, because if I want a specific order, let's say someone says, well, I just want a pink and blue alternating necklace, I need those specific instructions, and those instructions are in your DNA, and they're going to tell the ribosome, because remember, the ribosome is going to be making proteins. It's going to tell the ribosome which beads to put together in which order. That is called the primary sequence. So our amino acids will be linked together using that dehydration reaction. And we're going to form a covalent bond between them, but we call that, we kind of give it a proper name. That name between our amino acids is called a peptide bond. When we have a peptide chain, we're going to get two or more amino acids linked together. And eventually we have what's called a polypeptide, which is many amino acids strung together. We tend to call proteins, we also call them polypeptides. Here is our dehydration reaction between our two amino acids. And you can see as we pull out that molecule of water and we hook up that covalent bond, that specific covalent bond is going to be called a peptide bond because it's between, we used to call amino acids peptide residues. That's where this nomenclature came from. So two peptide residues, peptide bond, polypeptide. Now we call them amino acids, and we haven't really changed the rest of the names. When we have our protein structure, so like I said, we have our primary sequence, which is just how the beads are put together, how they're strung together. Based on those side chains, we can get different shapes. We can get what's called an alpha helix or a beta pleated sheet, and that's referred to as our secondary structure. Right, so our secondary structure could be either coiled in an alpha helix, or I always call like this one would be like if you use your curling iron, your ringlets, and if you use a crimp iron, you're going to have your crimper. And that's kind of what this looks like, that beta pleated sheet. And then that's going to all fold up into this globular shape. And that is our tertiary. And so in our tertiary structure, you could have alpha helixes, you could have beta pleated sheets, you may have more than one, you may have 2, 3, however, whatever combination, depending on that primary sequence. Now, not all proteins have what's called a quaternary structure. A quaternary structure is where more than one protein comes together and snaps together to form a functioning unit. Sometimes like hemoglobin has 4 pieces that snap together. Our insulin receptor has 2 pieces that snap together. And so some proteins are just one piece, right? So not all proteins have a quaternary structure. So here's a picture of that primary structure. You can see all the colors and the different beads put together. Our peptide bonds are put together. Here's a close-up of our beta pleated sheet and our alpha helix. And again, that's very dependent on those side chains because they're going to form those hydrogen bonds. And how they form those hydrogen bonds are going to give us our secondary structure. And some areas don't have a secondary structure at all. That just they don't have that uniformity, I guess you could say, in their hydrogen bonds. And so then that tertiary structure, we can see here, sometimes you can get a positive and you can get a negative amino acids coming together. You can get a non-polar region forming like a pocket to stay away from the water. We can get two amino acids that have sulfur and they're forming what's called a disulfide linkage. And then here's another one where just hydrogen bonding. So those different types of interactions will fold up into that globular shape, that 3-dimensional shape that is so important in giving it its structure, its overall structure. And then here is that quaternary where I said some proteins need more than one subunit to function. Here is our hemoglobin. You can see we have an alpha 1, alpha 2, beta 2, and beta 1. And so we have these 4 different groups that are coming together to form hemoglobin, which is the protein that carries oxygen around in your blood. Now, denaturation is a process in which we unfold our protein. So like I said, each protein has a very specific structural shape to it. And I always call it like my paper clip. If I have a paper clip, it has a very specific shape to it. And as long as my paper clip has that shape, it can do its job. It can hold papers together. But if there's a change in temperature or pH or salinity, that's the salt amount, or other chemicals, we could unfold our protein, and that's called denaturation. And now my poor paperclip, it's not going to work anymore. Okay. In some cases, we can refold those proteins. A lot of times we have extra proteins, which are called chaperone proteins, that will help to refold. But as you know, if you've ever tried to refold a paperclip, it just does not work. And many proteins, that is what happens. And that's what happens when you get a fever. If you get a fever and it's too high, you start denaturing proteins and it can kill you. And we can't, oh, see, oh, I broke my paperclip. So not good. Okay. So especially when you're talking about starting to denature proteins in the brain, which is very sensitive to all kinds of different types of changes in its environment. Your brain is very sensitive. So like I said, denaturation can be irreversible. For instance, I don't know how many of you guys like your eggs over easy. And if you ever go too far, you can't change it. You got to start over from scratch. I don't like my yellows semi-hard. Nope. All right. So, and that's what's happening is you're denaturing proteins. When you cook food, you're denaturing those proteins. And the idea is to make it more easier to chew and to digest. And so with that, you can see there's actually a color change. A lot of times when we have chemical reactions, one of the indicators is a color change. And so we have this color change here with your egg whites that are clear going to white. And so that is a good example of denaturation. We denatured the egg. Very critical, like I said. If you can't refold it, the protein will be broken down and recycled. Like I said, there's some times where we can get some assistance with our chaperones and refold some of those proteins. And then we always try to give you some real life examples of where proteins aren't working properly. And there is a protein that's associated with the plasma membrane. It is actually what we call a channel protein. We'll talk about those a little later. And it allows for proper salt balance in your cells. And there is a genetic disorder called cystic fibrosis, where that protein is not made properly. And so what happens is it affects a lot of different organs. It really affects the lungs, but it also affects the pancreas, liver, kidneys, and intestines. And so what happens is, like I said, it doesn't transport salt and water properly. And so you end up getting this mucus that builds up. We've actually found out that in the DNA, we had one small error in one nucleotide, which we'll talk about nucleotides in another lesson. One nucleotide is different. So one wrong bead. So on my pink and blue, I got a purple in there. And that purple caused that protein to not fold properly. It was in a critical location. The protein didn't fold properly. It can't do its job. And this is life-threatening for people that have cystic fibrosis. It is a life-threatening genetic disorder. So here is a picture of that channel protein. It's not working properly. And so we end up getting this mucus coating. That mucus coating sticks to the lungs and interferes with being able to breathe. So in the lungs, like I said, that altered mucus provides an environment. So not only does it prevent breathing, but it also sets up this lovely habitat for pathogens, for bacterial infections. And so people that have cystic fibrosis, this is a constant battle with them. They're on a lot of different medications to help with that mucus buildup and to prevent this potential of infections. So this is one of the things that will, eventually lead to them passing. Also, like I said, it also gums up their pancreas, and they have a lot of trouble digesting their food because they can't get the proper enzymes into their digestive system to break down their food that they eat, which now we give them digestive enzymes. So every time they eat something, they have to take digestive enzymes with it. So that is the end of our lesson with proteins. I hope you found that helpful. And if you have any questions, and hopefully you can relate this to the lab that we did. Remember, we did our BioUret test, or you will be doing your BioUret test, depending where you're at in this course, in which we will be doing a test that specifically tests for that peptide bond. All right, so I will see you in my next presentation. And like I said, I hope you found that helpful. If not, please ask me for help. All right, bye.